from Microbe TV, this is Immune.
Hello, I'm Stephanie Langle, and you are listening to The Immune, the podcast about the body's defenders against disease. Today, joining me from Ithaca, New York, we have Cindy Leifer.
Hi. Welcome back, everyone. It's great to see you. Great to see you, Steph, coming back.
Yes,
How's
and
the
I don't
baby?
know if it was brought up, but I was away on maternity leave. You all did excellent jobs with the past three or four episodes. I've been listening, and it's been wonderful to keep up. So, bravo.
We're great. We're glad to have you back.
Yes, it's great to be back. And from Madison, New Jersey, we have Brianne Barker.
Hi, great to be here. Great to see you back, Steph. We're excited that you're here, and I'm excited to have another episode of Immune.
Wonderful, wonderful. So we don't have Vincent today. He is visiting another university, so it will be us three, and we are going to be interviewing a very exciting guest. And I'm going to introduce her. She is from NYU Langone School of Medicine. She's an assistant professor, Carla Nowosad. Welcome to Immune.
Thank you very much for having me. I'm very excited to be here.
Excellent. And I have been following Carla's work for quite some time now. I am somebody who would consider themselves a B-cell enthusiast. I do work, more on vaccine responses and infection responses, but Carla really digs in to the mechanisms of what is happening in the Germinal Center, which she's going to share all about for us. And we're excited to have her on today. I think, like our other guests, we really want to know about your background. Where did you grow up? How did you first get into science?
So I grew up in the UK in an area called Suffolk. It's kind of countryside-y, lots of farmland and similar. And I really just got into science because I loved it. I don't know. I didn't have any kind of eureka moment except for my mum driving me around our hometown saying, "Do you really think you should apply to med school?" And me being like, "I thought you wanted me to apply." And she was like, "No, we don't care." So I was like, "Oh yeah, no, I don't want to apply." So I think moving away from that stereotypical med school, if you like science path, and looking into the other alternatives meant that I ended up studying biomedical science, and it all started from there, really.
That's great.
So were you interested in science from really little, or were there moments that you realized that maybe research was more what you wanted to do, and that kind of was released after you were released from the shackles of applying to medical school?
Yeah, I mean, I had been doing kind of some extracurricular science all the way through my schooling. So I was really lucky. In the UK, we're really lucky. I was picked to go to university campuses and do science days. And I was in several gifted and talented programs where we could go and do lab work. So I already knew that I was going to be pursuing something like that. But I mean, I was like one of those annoying kids. Like I was doing lots of sport, I was painting, I was doing this, that, and the other. So I was interested in a lot of different things. But I think when it became time to really apply to university programs, I got a bit more serious and decided it was going to be biology for sure. And at that point, thank God I didn't get in. I was going to do neuroscience. I was going to do like an integrated master's in neuroscience. That's what I was really, really intent on doing. And for some reason, I even did get in. For some reason, I didn't do it, and I just did biomedical science. And it was a great decision because when I started learning about immunology, that's when everything changed, and I was like, yeah, this is what I'm going to be doing.
Where were you at university?
I was at Warwick University, which I think is not really a classic kind of research school, but I just loved it. I absolutely loved it. The best thing about Warwick is that they had this great program where you could take a year off and go and work in industry.
Oh, wow.
So then I worked at a monoclonal antibody company.
Oh, wow.
Yeah, and that's where I was interacting with people with PhDs. We were making monoclonals using, at the time, it was kind of like these methods where we would isolate single B cells and use this fluorescent technique to pick the ones producing the good antibodies. And I was able to make a monoclonal in a year there. And I was just absolutely sure I wanted to study plasma cells. And I was like,
The
yeah.
rest is history.
Yeah, and I was like, it's cool. I'm going to go back to work, just get the degree, go and do a PhD, and then I'll come back to this drug company and work here. So I think I would, yeah, and then I started doing a PhD, and again, everything changed. But yeah, that was the turnaround experience where people I was working with there were that I just thought they were so amazing. They were really smart, and we're still in touch. Actually, it's great. So I really had a pivotal experience there where I was like, wow, this is like people's jobs, where they like read and figure out new things to do, and then they make something and it's a drug, and that's what happens. So yeah, I loved it.
That's great. Was that program in your university specific to that university, or does the UK do this model where you go out and you have an internship and then you?
Yeah, it's common. It's common. So at the drug company I went to, there were probably 8 of us, and we came from different schools.
Oh, very cool.
Yeah. So we already had like kind of a teeny tiny little network then of people who were interested in getting onto PhD programs or working in industry all around kind of antibody production and drug production. So yeah, it was a great experience.
Sounds
Really
like we
good.
could
And I
learn
feel
a
like
thing or two from that as far as PhD programs here in the US.
100%. I mean, it was paid. I was like completely broke. So I was happy that this was paid. I was like living the high life, I felt, in my third year of uni. We lived in a house with the other students. And yeah, I think it was a great experience. And it kind of didn't extend things too much longer.
Right.
And it gave you a real idea of what, I mean, obviously what I do now is different, but it gave me a real idea of you're going to need this PhD if you want to have a career in academic science. Yeah. And everyone, it was like abundantly clear. Everyone in decision-making capacities had completed a PhD, and everyone else was working more in assay development and the kind of day-to-day running of things. So that was the kind of crossroads where it was like, do you want to be in this more active decision-making capacity or not? And if you do, you need to start getting serious with the last year at college, making sure you get on the right PhD program and thinking about the kind of mentors that you want to work with in that phase. So I went back very motivated.
Right. And having those connections. I mean, if you did not pursue academia, you have industry connections. That's perfect.
Yeah. Yep. And I see them still all the time at conferences. It's crazy. Yeah. It's really good.
Small world. And then after that, you transitioned to your PhD. What program did you, I mean, we know who your PI was, but what made you choose that lab? What was that process like for you?
Well, a series of lucky decisions, I think. So I went on to do my PhD at what is now called the Francis Crick Institute, but back then was called the National Institute for Medical Research at Mill Hill. So back then it was split into these 2 campuses. One was in central London and one was in North London. And it was just this crazy place. It was shaped like an antibody. Can you believe? It was like, it was shaped like an antibody. So there was like a central kind of bar shape. And on each edge were these 2 corridors that were like the little VDJ.
I was going to say, the way you're describing it makes it sound like a dimeric IgA.
It was an IgA.
Okay, I love it.
Can you believe that? Yeah, it was an IgA. So Mill Hill was just the most it was like a mythical place, you know. There were not many labs there. Everyone knew each other. There was a bar on site. We lived on site.
Is it funded through the government? Is it federal?
It was through the through the MRC, Medical Research Council.
Right. Okay.
Yeah. So they took very few PhD students. So we knew the whole cohort and we knew each other all the way through, you know. It wasn't that we knew each other, picked labs and then didn't keep in touch. We were like very, very close. So it was really an amazing experience to be there. And I'd never heard of it, of course, because I didn't know anything about academia. I just knew I'd been at this drug company, so I needed a PhD. And I needed to go somewhere good because then I would have the best opportunity. So I was sort of, I don't know, I feel like when you're newer to academia and you find it during your undergrad, these are the metrics you use. Like, where's the most reputable place? Okay, I'll try and go there. Does anyone do B cells? I'll try and go there. So I sort of like ranked them top to bottom and applied to, I had a very inflated sense of what I could achieve at the time. It's a good strategy.
And
I
you
was
need
like,
that
go
though.
high.
Just
Yeah.
try it, because the worst case scenario, okay, you don't get it. There's going to be other
Yeah.
opportunities. You
I
got to
truly
try.
had no idea, and therefore no fear. So I was just going in saying, yeah, I should be on your program. Why not? So the one that I really liked was Mill Hill. And a bit like here, the better programs accept early. And so I had a place there, and I just thought, yeah, sounds good. And it was really weird. The interview process, I remember it so clearly. It was like X Factor. On the end of day one, they split you half and half after dinner and said, you guys are finished with the process, and you guys are coming back the next morning.
Brutal.
And then you would come the next the next
Whoa.
morning
Serious?
and do your panel interview where you had to present a paper. And mine was the one where with Crelox they floxed out the BCR.
Oh, cool.
But like I didn't have much research experience, so I spent my whole time saying, can you believe they can just delete things from cells? And everyone on the panel was laughing, and I was
That's
like,
so
okay.
cute.
And I was like, can you imagine what you could do with this? This is unbelievable. So
Oh,
yeah,
that's
I
awesome.
think they thought I was very sweet and very stupid and naive. But I had some like serious research experience at that point. I had a paper. We'd made a therapeutic monoclonal. And yeah, so yeah, I think it was after I'd got past the amazement of Crelox, I was able to say kind of clearly what I thought this would be good for, and I knew really clearly that I wanted to work on antibody production, B cells. I clearly knew at that point that I wanted to stay in academic science, because they were asking all these, what are you going to do in 5-year questions? And I was like, probably a postdoc in the US. So I already had some ideas about moving through the system. So yeah, for some stroke of luck, they thought that was charming and not just ridiculous. And I got on that program, and yeah, I went to work with Pavel. Another kind of lucky, very good decision, because he's a really good guy, an amazing scientist. And we just had, this dream PhD situation where it was the end of the first year. We had a phenotype, and we were putting it together.
Amazing.
Wow.
So can you tell us a little bit about what you worked on in that PhD?
Yeah, so I went to work with Pavel Toler, who had just opened the lab at the time at Mill Hill, and he'd come from Sue Pierce at the NIH, and he was doing this work looking at synapse formation, immune synapse formation. So using bilayers mostly, and then the first PhD student in the lab had developed a system where we could look at B cells internalizing antigen. So instead of using lipids, she was using whole cells, sonicating them to cut them in half, loading them up with antigens, putting B cells on top, and watching the B cell receptor in real time move that antigen around and also rip it off. And it was really amazing work. And we started the project being like, which B cells do this differently? So we were putting lots of, we wanted to look at anergic B cells to start with, because that was the cell du jour at the time, to be like, these amazing B cells that don't do anything and they're silenced. So we were looking for anergic B cells. These were very difficult to find for one reason or another. I'll speculate on whether we think that they're abundant in tissues, but what we ended up doing was looking at germinal center B cells. And we saw that they just, my favorite thing about microscopy is you see straight away if something is different. And we could see straight away these were making really weird synapses, very, very different in their structure. And the immune synapse is a pretty common structure between B and T cells. So to see a departure from the very common bullseye structure of the B cell receptor, it was very striking. So we just chased it very hard to see what was going on in the germinal center.
So what actually looked different? Sorry.
Yeah, no, go on. So how a synapse is normally structured is the B cell receptor touches antigen, clusters to make this big cluster in the middle of the contact, and it's surrounded by all of these scaffolding molecules which keep the two cells together for enough time that those receptors can signal and internalize antigen. Then the B cell's activated and it goes on to live its life and produce antibody. In the germinal center, we have to remember B cells have already been activated. So that's kind of the first difference. And we saw that when we put them on these, firstly, bilayers, they would move antigen around. And instead of pulling it into the center, they'd sort of actively push it to the edges in these teeny tiny little clusters. And when we first saw this happening statically, we thought it's okay. They fail to centralize. They start and then they stop because for some reason they can't do it.
Right.
And then when you image it live, you start to see these clusters actively being pushed out. So the cell is trying not to let these clusters touch each other.
Right.
And we just thought it was so cool. And then started a long and winding path of figuring out why would the cell want to do this? Surely then doesn't touch very well and it doesn't stay together very well. And we started unraveling that probably in the germinal center, you want to tell if a cell is not very good by giving it maximum chance to fall off another cell. And if it can overcome these higher thresholds and higher barriers, maybe it's the best cell. So here you're trying to tell the difference, tiny differences probably over time that the cells are making. And we think that this is probably to do with that. And we were able to show through using these wound up little pieces of DNA that actually the B cell receptor can pull harder in germinal centers rather than elsewhere. And this could be because FDCs are stiffer.
Right.
So there's these multiple layers of regulation that happen beyond just the B cell receptor binding better.
Right. And for our audience, FDCs are follicular dendritic cells, which are involved in this process. And so it is amazing. You're finding this regulatory way of affinity discrimination.
Yeah, absolutely.
A mechanical. It's really interesting because we often think about cellular, intercellular mechanisms, but the physical nature of where this antigen is being presented and how is really amazing.
Yeah, and there's a few important points to consider. That antigen doesn't float around your body very much.
Right, right.
Most of it gets scooped up by other cells, other molecules, and then the way that it gets handed off and makes its way to a B cell is integrally important to how that B cell perceives it and becomes activated. So the space element is really, really important. And that sort of sparked a long-term interest in understanding where things are spatially, whether it's where the receptor is in the surface of the cell at any one time, whether it's where those cells are in the organ. So for the germinal center in general, of massive importance, where the cells are, how they get there, how they move. And that's what I study now, how cells decide where to be where they are.
So I may be getting ahead of us a little bit, but based on some of what you just mentioned, did you see the same thing happening kind of in different germinal centers in the same secondary lymphoid organ or in different secondary lymphoid organs? Is this a general thing that you see in germinal centers, or is it specific to some specific ones?
Different. Yeah.
Very good question. Very good question. So what we were doing at the time, because it was technically challenging to do these kinds of studies, and these cells, germinal center B cells, are so fussy. They are ready to drop down dead because they really are. You're making these dangerous mutations in cells, and if anything's untoward, these cells die really quickly. So they're sort of on the brink of death, ready to be saved by signaling. So if you take these cells out of their environment, they're super finicky. So it took us a significant amount of effort to get cells out in the right number to do these assays. And what we were doing at the time is we were immunizing the spleen with something quite generic, like sheep red blood cells, just to generate large numbers of germinal centers. So what we figured out with Pavel was a generic mechanism that we were seeing in regular germinal centers. Whether this holds true in some of the more unusual germinal centers that we've become interested in more recently is TBD, So we're spending a lot of effort figuring out now whether the rules shift one way or another. If you're a germinal center that was induced under autoinflammatory conditions, whether we can fiddle around with the way that you were immunized and this changes things, and really what that actually means for the output, which is not usually what we study when we look at germinal centers, but it's ultimately what we're interested in. How does this structure change the antibody? Really what we're studying is how do we drive antibody responses, and where are the points where these vary, and where can we intervene? So the long answer to your very good question is we are not sure. Yeah, things get exponentially more complicated as we go to smaller organs.
Right. Because you're trying to, again, if they're finicky coming out of the organ and you're trying to get a certain number of cells for your experiments.
Yeah. And you
Right.
can imagine as we narrow this down to say, what about a different isotype? What
So,
about
yeah.
this one versus this one? What about this location versus this location? Then we have to really sharpen up our technical prowess and think about ways that we can either miniaturize or optimize to really recover the maximum number of cells to do these kinds of assays.
Sure.
The real strength of what we did with Pavel was to be able to look at thousands of cells interacting, and this was like subcellular resolution at once. So we could analyze thousands of cells like flow, but with the imaging link.
Right.
And that was really what allowed us to uncover this heterogeneity that was completely hidden by other techniques.
Right.
So we want to retain the resolution, but yeah, it starts to become technically challenging, but we're so close to answering your question.
Oh, that's
Next
amazing.
time, maybe.
Yeah, maybe what we'll do is we'll do a part 2 in a year if you have
things Carla said she was going to do. Did she manage to do them?
Well, that's great. So this is your PhD, and what's neat to see, I mean, I don't know if it's neat or not, it's just different. There's only 3 authors on that Nature Immunology paper, yourself, your advisor, and one other. It's just rare, often you see many authors.
Yeah, it was really kind of, I don't know. Obviously it was the only experience I had. So
Sure.
it was something where like Pavel, he's just really insightful anyway. He had a really clear vision. And we did it, and it worked. And I think I can safely say it doesn't normally happen like that.
Yeah, for our audience, that's a great story, but often not common, right?
Yeah, and there was definitely like a muddy part in the middle where we couldn't figure out how these were different.
Right.
But to have a great phenotype is a dream situation for a PhD student, to try something and be like, oh my God, they look completely different.
Mm-hmm.
That's kind of what you're hoping for. So yeah, we were very fortunate. And the other author, Caitlin, was an expert in constructing these force sensors. She was a biophysicist. So we had a few different minds working on this.
Right.
And we could really get to the biophysics of how these cells move things. Yeah, it was cool. It was a good experience.
Very cool. So then after your PhD, you had already, it sounds like in your interview of your PhD, described that you'd like to take that step to the postdoc in the US specifically. So tell us about that journey. Now you're moving, countries. What was your next, how did you select your postdoc?
Well, my paper was accepted with Pavel, and we were moving from North London to Central London to this new institute.
Wow.
And he'd already hired a bunch of new postdocs, and he said, okay, it's time for you to fly the nest. And I was like, no, I thought we were going to have another few years. And then I started looking around, and I guess the thought process was, do I like these single molecule measurements, or do I like the Germinal center? Because I could have gone in 2 directions and kind of deep dived into these new techniques, or kind of really tried to figure out what was going on in the GC. And I sort of picked my favorite few people working in each area, reached out, visited. And at the time, Gabrielle Victoria had just published this. I mean, people must know about it now. It's kind of older, but had just published about confetti,
right?
Otherwise known as Brainbow. And they were looking at clonality of germinal center B cells. And it was just one of those papers where everyone was like, whoa.
Yep. Look how cool this is. Yeah.
It's really
we talked to him.
Beautiful.
We did.
Do we know which episode that was?
I don't remember exactly.
We can
episode.
direct
last year.
I'm
Googling.
Listening to Gabrielle talk about germinal centers is quite something, isn't it? It's amazing.
Yeah.
It's
he really can take you through the whole history, and it's really incredible.
Totally. And he's very understated in the way he describes his contributions. So it's
absolutely.
So I was sort of traveling around, talking to people who were really, really expert in the Germinal Center. And it was like the month before I defended. So it was like really, I was like sitting with Jason's sister talking about my work, and it was more thorough than my actual defense. It was really amazing. So I came back from that trip to the US being like, oh my goodness. And then my real defense was a breeze. So I had like a pre-defense by talking with Gabrielle, Jason, all these other people. Yeah. So I'd been going around talking to the GC experts, talking to a few people who were cytoskeleton experts.
Right.
And I think when I was with Gabrielle, I was like, yeah, this feels right. And then another chance encounter, someone from the PhD program who graduated a few years earlier was working at Rockefeller and said, you should talk to Daniel Musida. He's doing this wonderful imaging, and he's got a great lab, and I think you'd like him. So I stopped by Rockefeller. Daniel was writing an R01, I remember, and he was like, I don't have time, I'm too stressed, but we can have lunch. So we had lunch, and he said, Daniel's a real straight shooter. He's great to talk to. And he said, listen, Gabriel's going to offer you the position. So I don't want they're obviously good friends. So he was like, I don't want there to be any weirdness. You can either do something with us together where we could start looking at the mucosa. Daniel's a mucosal immunologist,
right?
But who works on T cells mostly. So he was like, you can look at B cells in the gut. We've been wanting to do it for so long. You could be great for this, or you can work with him, no pressure. So I got this what I thought was this amazing once in a lifetime opportunity to take a position with both of them. I was really, if any of you have been to New York, you might understand that I was very excited by the idea of being in New York. I stepped off the like filthy subway at 10 and I was like, this is it. I'm home.
I'm here. I've arrived.
I was just like completely overwhelmed and couldn't get a cab, but I thought, yeah, this is it. So yeah, like lots of things came together and I thought, yeah, this is still Germinal Center, but it's going to be a new challenge. That interview process taught me like I really wanted to work on the Germinal Center more than I wanted to work on single molecule techniques.
Right.
So it's more kind of biologically minded than the physics side of things.
Which is an interesting distinction, I think, for people who are looking towards a postdoc. I think those are two different strategies you can take.
Yeah.
You can be so obsessed with something that you will be able to persevere through the challenges and you know that is the thing you want to study. But some people maybe don't have that thing, but they're interested in the technology. And a lot of times you can apply a technology to many different fields. So I'm thinking about structural biology, for example. You would maybe go learn that technique that could be applied. Yeah. So I don't know if Cindy or Brian have thoughts on that, but as a way to think about postdocs and how to diversify yourself, that's kind of like, there's many ways, but those are two.
Yeah, I think that one, like you said, one way is staying on track. You've had the same interest the whole time. I sort of took that path. But the other thing is, after you do a PhD, that postdoctoral time is also an opportunity to say, okay, I already know what I want to do, and I'm going to go pick up this one thing that's then going to enable me to ask this question that can't currently be asked. So yeah, I think that considering that as you're going through, thinking about what your long-term research interests are, and do you want to like continue down that path or maybe jag off of it to pick up something that gives you a unique niche going forward when you start your own lab.
You're not married to anything.
Nope.
Even though you now have a PhD, sometimes I think students can feel pigeonholed into being like, well, I'm now an expert in this. And it's like, no, you just got better at learning quicker.
Exactly. That's the best time really to switch. It can sometimes be harder the longer you go, to a postdoc.
Yeah.
To be honest.
Honestly, I think that you guys all said what I think better than I would have said it. So I think that all makes sense.
So you concur.
But I will point out that if people are excited to hear about confetti, they can hear about it on episode 52.
Oh, thanks for looking that up.
Thank you.
What was the name of that one with him?
B-Cell Boot Camp with Gabriel
B-Cell Boot
Victor.
Camp. Right.
It's good. Sounds serious.
Very appropriate. Yeah.
So I love this, though, the marrying of the mucosal immunology with the expertise and the tools to study the Germinal Center in a different way. And so frame for us the questions in the field, the drama, as you may say, in understanding what's going on in the gut and why it's different from a Germinal Center reaction outside of the gut.
Yeah, absolutely. So the intestine became interesting to us for a few different reasons. So firstly, there's several sites that B cells live in the intestine. We would normally think of a B cell living in a spleen or a lymph node, and the gut is innervated by no less than 8 lymph nodes, sometimes more. So it's a lot of lymph nodes, but actually there's a bunch of B cells also living in these structures called Peyer's patches, which are lymphoid follicles which are positioned on the edge of the intestine. And these have a unique structure of their own, which is like a whole different literature. But both of these locations host germinal centers all the time. The germinal center, for those who don't know, forms usually during inflammation, but the gut, you can either think of it as chronically inflamed or or tolerant or whichever, but nonetheless, germinal centers are always there. So there was the question in the gut was, what do the germinal centers do that form when you don't have infection? There was vast literature saying they're not important for anything. And actually, the antibody that forms in the steady state, which is grams, like multiple grams of IgA being secreted just in normal everyday life right now as you're sitting, and that IgA was all T cell independent, and majority was non-specific or polyreactive. And this was just sticking to bacteria like Velcro. Some of it because the bacteria is spiky, sometimes maybe because of mucus or other mechanisms. And actually, there was no specific mechanism to select antibodies that actually bound to members of the microbiome. So this was a massive open question in the field. I think what really got me and Gabriel like confused, I suppose, was the notion that the microbiome is exquisitely regulated. It doesn't go wrong normally. Right. But the fact that this was happening just because of random antibodies sticking to it that were non-specific, it doesn't resonate with people who have spent time studying antibody dynamics. Or the germinal center. The germinal center doesn't select antibodies that are no good normally. It selects mostly antibodies that are driven towards a certain outcome, high affinity normally.
Right.
And it's really weird to think that germinal centers are there all the time, but they're not doing anything, or they're not selecting effectively, and actually they're sort of just outputting clones which do all sorts of things. But these things don't make you autoimmune normally, but they do regulate the microbiome. So there was some things in our mind that we just couldn't get our head around, even though
and there
the data there.
Some literature in germ-free mice that would still have germinal centers. And so
there's a bunch of literature which is hard to understand. So germ-free mice have germinal centers, absolutely. But also if you take away T cells, you still have IgA.
Right.
So that data became confusing, but you lose germinal centers if you lose T cells or T cell help or any of the normal reasons you would lose germinal centers. But you kept IgA and the microbiome wasn't that dysregulated. So that was the argument for germinal centers not really contributing to the IgA that regulated the microbiome. So it was really an open question. And we had confetti and we had mice with intestines. So that's where the project started, to say, okay, if we look at selection without taking away the microbiome initially, without manipulating this in any way by taking cell populations away, would we be able to observe any natural selection processes in our B cell repertoires? And we were looking in both the mesenteric lymph nodes and the Peyer's patches. And we could see pretty clearly early on that some germinal centers here were very strongly selected. And by selected, I mean that a single B cell clone had made mutations that were so effective that that clone proliferated to take over the entire germinal center. Something that we know is important in immunization responses to promote high affinity antibody responses.
And that's not consistent at all with what was the going trend of these were non-specific and they really didn't play a role and they just pumped out antibody that didn't fit with the model.
Yeah, but there were some hints that maybe this wasn't the whole story. So if you looked at humans, all of the plasma cells in humans, which are the cells that actually produce the antibody, which live in the lamina propria, another location to make things more complicated. If we look at those plasma cells in humans that are IgA positive, all of them are mutated.
Right.
And it's currently accepted that mutations occur within or just before germinal center reactions, at least in activated B cells, which had T cell help. So unless we could take a real departure from what we know about B cells and say that actually mutations can occur at similar rates outside of germinal centers, the logic in humans is that these germinal centers were producing most of the IgA positive plasma cells that were in the intestine at any one time.
Right. And what's neat about the technique that you use, which if those who are listening haven't listened to Gabrielle's episode, is that you can see this clonal burst, which will have you describe visually by the confetti imaging. And it's a beautiful thing to see.
Yeah, there are some complications with the gut, but by and large, it worked just as it would work normally. So normally we would start an immune reaction, label it in 10 different colors. And if the cells had selected, there would be less diversity in the colors after selection than before, because one of the colorful cells would be selected and expanded, and that color would be overrepresented. And if you look at it in a lymph node, this would look like one germinal center being, say, red, rather than being all 10 colors. So in the gut, the main problem we had was germinal centers are there the whole time. So if we color a germinal center that had already formed and selected, we would assign one clone 10 colors. So the mathematics of confetti get quite complicated when you don't have a start point. So what we had to do was just label everything, wait for a period of time and say, we're only going to look at the ones that look selected. All of the rest that don't look selected, we can't truly tell whether they are selected or not selected. But to make things simpler, we're going to only pick the ones that were visibly selected.
Mm-hmm.
To answer the question, and do any of these structures select towards the microbiome?
Right. And then you're able to take those cells and single cell sequence them. I imagine that's a difficult technique. You have your vibrotome and you're trying to slice these lymph nodes and extract single cells. Was that a technical
it's easier to describe than to
achievement?
Do, I think. Yeah. It's easier to describe than to do.
How do you deal with the sort of cell number problems at this point?
Yeah. We're pretty good at recovering very, very small numbers of cells. I think what we do is we try and think about how many cells we actually need sequences for. Mm-hmm. And if it's one plate of cells, that is 96 cells. So that's actually not that many cells.
Sure.
So we just get very good at saying to the people running our sorts, no, there are cells there, just keep running. Because they'll be like, this sample's rubbish. And you're like, no, keep running. There's going to be cells. So yeah, it's like a nail biting sort. But so we image the lymph node, we slice it with a vibrotome like salami. We re-image to find our germinal center that we like. And then under a light microscope with a razor blade, we cut just the half of the slice with the germinal center. And then we just touch it with a pair of tweezers and put it in a Eppendorf tube, and I hope to God that there's a pellet there.
Wow.
Yeah, it's really technical and it's really heartbreaking when you saw a really good one and you don't know what happened to it. But with a bit of practice, I think it's easy to get good at it. And
so you have the clue by looking at your different germinal centers. If they all turn one color, there's selection, and then you can take those cells, sequence them, and make the antibody that clone would produce. And that was your next clue. The data that you generated from that is they were specific. They were antigen specific, affinity matured. Right.
Yeah. So we could produce the actual antibody that the B cell would have made, because the B cell receptor is this molecular blueprint, which is useful for us. And we could do a few different things. We could figure out the evolutionary history of the clone, which is very interesting because it really tells us if this is antigen driven. If the mutations are happening logically down a tree, it really is strong evidence for antigen driven selection rather than random mutation. So that was our first hint that these clonal bursts, these events that we know are very, very important for immunization responses, was happening in the gut. Right. The cool thing in the gut was it was happening again and again. We would find these clonal bursts all the way through the lifetime of the clone. So this was kind of telling us that these germinal centers maybe are old, and maybe different variants of the clone burst over time. Very cool. We hadn't seen a lot like it before.
Turning over completely, and that's the thought of how these clones are then they pop up in another germinal center. They survive and.
Well, yes and no. So others have shown that they can find clones throughout Peyer's patches. For us, with the data we got, we can't say that because it's very difficult for us to find these clones elsewhere unless we sequence the whole thing.
Right.
What we do see is if we label with confetti and leave for a long period of time, cells do begin to turn over. So those germinal centers will go back to being black. But actually, if you look at really, really late time points, maybe 12 weeks, some germinal centers stay colorful. And weirdly, and we're really interested in this now actually, weirdly, the ones that stay colorful seem to be the ones that were selected.
Interesting.
So if you became monoclonal or dominated by one clone, you seem to somehow be resistant to turnover. And we truly don't understand the dynamics of this at all.
And so pro-survival signals potentially that we don't understand is what's driving this within that center.
One option is that. The other option is they're resistant to invasion by new clones because of some competitive entry requirement.
Got it.
Met by higher affinity, perhaps, similar to the notions of antibody feedback and these other kind of, not theories, I guess they have a lot of evidence, but these other mechanisms that are in place during immunization.
When you were looking at this in response to microbiome, were there any particular antigens that you saw that were particularly important? Or were it kind of all sorts of antigens?
Yeah, good question. So it started to become really, I mean, people, when we told them about this project, were like, this is impossible. And we were like, no. And then, yes, it was very difficult. So when we got these clones that were selected, we could identify that they bound the microbiome in mixed samples. And when we tried to figure out exactly which bug it was, it started to become very, very difficult to narrow down from the thousands it could be to the one it was. We got pretty far in trying to do very, very low input sequencing of the bacteria that we could sort from the flow, but it gets very difficult not to contaminate that and not to just pull down other bugs that are nearby. So to get around the problem, we decided to use mice that only had 12 bacteria.
Oh, right.
Therefore, we could see if there was selection, pull out the single color clones, produce them, and then we could say which one of the 12 did you bind. And we figured this would be a much easier way to figure out if there was such a thing as a driver bacteria, or if any bacteria in the right time or place would be able to cause this kind of reaction. I would say that also wasn't as easy as it looked. And you really have to produce hundreds and thousands of clones to make any robust conclusions. But I will say of the clones that we got from those reduced complexity microbiomes, they bind different bacteria.
Right.
Not always and not exclusively, but generally. And if you exhibit this really strong selection, so far it seems like that is towards a single bacteria. But we're not ruling out the possibility at all that these could be to common epitopes that exist on many bacteria, or that there's such a thing as breadth selection, or there's other things. So we don't know if in the germinal centers from the gut, there are loads of bugs, the antigens from loads of bugs, or if one bug deposits a lot of antigen on FDCs and causes selection that way.
And going from the extremely complex total microbiome down to 12 microbes, and then when you went further and took them all out, did you find anything unusual or unexpected?
Loads of weird stuff, yeah. So when we completely removed the microbiome, well, firstly, I'd never worked in the gut before. I thought, we'll take the bugs away and the germinal centers will be gone. Yeah. Turns out they are very much not gone, and everyone knew except us. So we were like, wow, this is so amazing. And then everyone was like, yeah, they don't go. So yeah.
But isn't that surprising? I mean, what else are they reacting to? Is it food?
Yeah. So no. So even more surprising is there's no IgA, I should say. So there's no plasma cells. They are gone. So what was surprising is when we used confetti and germ-free, we would have hypothesized that we remove the bugs, the thing that we think is driving selection. So these germinal centers will probably not select. However, when we take away the bacteria, every germinal center selects.
I was going to say they selected more, right?
More, so much more than anything we'd ever seen. To the point where we were like, whoa, wow, this is crazy. We've never seen such strong selection. So we would pull those out, sequence them. And we even did like much higher scale sequencing, not single germinal center. And we start to see the same clones popping up again and again. We do not only see the same clones in the germinal centers from one mouse, we start to see them in germinal centers from every mouse.
Oh yeah. So this like public
Peyer's patches.
Clonotype.
Peyer's patches, we start to see them in mesenteric lymph nodes. We start to question our sanity, to ask if we're just bad at pipetting. Did we just wipe plasmid everywhere? But these clones have very unique mutation patterns. So we have an internal control for our stupidity, thankfully. We can see each one is evolving individually. And whilst they share the same V or J, or sometimes the same looking CDR3s, they're not the same. They're definitely not the same. And in some cases, they don't even have the same J. We have some sort of clonotype variants. So these truly are individuals, but arising in different mice. And then we kind of got scooped, I guess, and another group found these as well. And it's sort of a heartbreak and a happy moment at the same time, where we're now very convinced that in germ-free animals, the repertoire is remarkably public. This is not like 70, 80% of the clones. It's a lower number of clones, but it's incredibly reproducible. The weird thing is as well, is if we use 12 bugs, we still see these clones to a lesser extent, but we still see them.
Interesting.
If we use a complicated mouse, which has hundreds of different bacteria, very rarely do we identify these. So now we start to build a picture saying, okay, no bugs, lots of selection, public clones, few bugs, intermediate selection, some public clones. The weird part about that is it seems to be on a per mouse basis. So some mice look SPF and some mice look germ-free. We don't understand that at all. Steph, you can probably help me think about this. I think maybe it's a pregnancy phenotype. Maybe it's a litter phenotype.
I would imagine so. Yeah.
Be super careful about tracking who came from what, but it was a really weird phenomenon. And then in the SPF, we sometimes find these clones very non-abundantly. So now we start to ask ourselves, when you become colonized with bugs, does it somehow reset the repertoire, moving from this selected public repertoire through to this complicated bug-specific repertoire? And that's really what we're trying to understand now, as well as understanding the obvious question of what do these clones do? What do they bind to?
I was going to ask, do we have an idea?
Yeah.
Is it self? Is it environmental? We don't know.
Yeah. So what we do know is it doesn't seem to be self, auto, or bug from our best efforts at binding. Also, these animals don't get overtly sick. So we don't think they're like hugely autoreactive in ways that we weren't expecting.
So no endogenous retroviruses. We did a B cell.
We haven't formally ruled it out, but that's our next step is the retrovirus and breast milk.
Oh, okay.
Yeah, because we wonder if these things are common between the two settings. The other thing is maybe this is developmentally encoded somehow, and it's not antigen driven at all. Even though these clones pass through germinal centers and seem to select, we're not sure if these are antigen driven even. So the mechanism is not clear, but we're looking into it in a number of different ways now to try and get a handle on what's going on.
And so can you speak to that again? So when you're saying breast milk, you're an antigen in breast milk of the mothers, you're seeing this in you're using young mice, you're seeing this in young. Are you only looking at germinal centers once they have weaned, and now you're going to kind of go back in time?
So
developmentally.
Yes, we are doing this. It's complicated because young mice don't have germinal centers.
Right.
Yeah. So our approach is to track every single day after birth when the first germinal center cells come, and to get those, every single one.
That's amazing.
Sequence them and ask, are you a public clone or are you not a public clone?
Right.
Very, very possible that they won't be public clones. Technically, this has been challenging, but we're really close to having a concrete answer for you. And if it's not a public clone, in SPF mice, what are the first clones that seed germinal centers? And then we can look pre and post weaning and see exactly how that overlap, how that reset happens. And if it's important to colonize in a certain order or a certain way. And most importantly, how this actually affects what's going on in the adult animal.
Right.
Whether this is actually a window of opportunity whereby if you gave, say, a human child the wrong treatment with, say, antibiotics, would this have a long-term output on their immune function in adulthood? Then that is something that's really worthwhile us knowing.
Absolutely.
Far more for just the fundamental biology of germinal centers, which I would love to study forever. A health outcome is something that we're really interested in.
Wow. And so the noise there's a noise happening with the commensal microbiota, and it does make sense evolutionarily, the fitness of the animal that you can redirect your multiple germinal centers to multiple different antigens. If you just had 12 public clones, you probably wouldn't last too long.
Right.
So that's very interesting. And then the other thing, I think I was reading your review on kind of like, you framed it, with space and time, place, space, and time of how these B cells operate. When a plasma cell moves into the lamina propria, the IgA and IgM B cell receptors are very much active, and they can bind antigen in that space. So now you have interaction with antigen, and we mostly think of plasma cells as they're just factories to pump out antibody, but likely they're reacting to antigen as well, secreting cytokines and doing things. So have you expanded out into looking at plasma cells? Or are you still thinking more on the germinal center B cell side?
Well, it's kind of that's a really good question. It's kind of Pavel's platform that we created when I was with him is perfect to look at this.
Yeah.
Putting the cells onto antigen on bilayers and seeing whether they form synapses. It's the perfect ex vivo system to see about the capability of signaling in these cells. But one can imagine that the germinal center outputs a plasma cell population that is a good reflection of the antigen that's in the gut lumen. But actually, they only get turned on if the antigen is aberrantly found in the gut lumen. So maybe actually the germinal center forms the kind of picture of what's going on in the lumen, and should that become dysregulated in some way, then the plasma cell would get turned on to intervene.
Right.
That's the model I like, but it's speculative, right? I just kind of
You're right,
like
because
to imagine it that way.
If the bacteria if it overruns the population, right, you would need to turn on that plasma cell to pump out IgA to calm it down, to harness.
Well, that comes back to the up and down. Like if one clone of bacteria becomes dysbiotic and overruns, then you can turn on those IgA cells and pull it back down, and it's just kind of constantly
but you want to be quick.
Yeah.
You want to be quick. If you had to do that through a germinal center, you're weeks behind.
Right.
Yeah,
right,
exactly.
So your germinal center is sort of providing the most recent snapshot of what's going on at the border. And then actually on location, these can tweak and dampen. That's how I like to think about it. But I would love to, I'd love to take the plasma cells out and look. I don't imagine we'd be alone at thinking that this is interesting to look at, Steph.
I don't think so. And so, one of the things I'm always thinking about is in the mammary gland, there is a population of plasma cells that have come from the blood that have we can track to the gut. And evolutionarily, I think it's because you want the antibodies that are derived from mom's gut ending up in the gut of the baby to harness its own microbiota. Correct. But what is really understudied, I mean, we know the chemokines responsible for this trafficking, but we know very little about what are the clones that are making it to the gland, to the mammary gland.
Right.
But it would help facilitate questions that you have about early germinal center development and then into adulthood. So we should, what grant can we write together?
We're trying to understand, like we've become more and more concerned, I suppose, because of our mouse husbandry and interested in what's going on in the mum.
Yeah.
So if you have a mouse strain that doesn't breed, for example, you put it on antibiotics.
Right.
Does this fundamentally dysregulate your immune populations in the pups? And then are these good models to study?
Right. Right.
So these are things we think about very often when we're starting to knock out all kinds of things in the immune repertoire, and our mice are struggling to breed. So then we have to think about interventions that don't disturb the microbiome, and that's sort of opened up a can of worms on how behavior during pregnancy, even during pregnancy, imprints then on how effectively you're going to become colonized, how effectively your germinal centers are set up, and how resettable this actually is later. So if I delay weaning of a mouse, we already know the Peyer's patches look dysregulated right into adulthood. But in terms of which clone, which bug, how this affects selection, it's very understudied.
It's fascinating. Wow.
Yeah.
I was going to say it harkens back to your title of your Nature paper, The Tunable Dynamics of B cell Selection. So I love that. Tunable dynamics.
Yeah, but figuring out how to turn the dial is really what we're interested in. Figuring out is there such a thing as a regulatory antibody? And if there is such a thing as a regulatory antibody that doesn't just clear things, what do we need to do to get those clones in and through a germinal center should we want to produce antibodies like that? Right. That's kind of interesting. I mean, I like what we were talking about plasma cells, but it's also worth remembering that in the gut, the antibodies produced there do not just clear bacteria.
Oh,
There's
for sure.
sort of a wide branching research literature about other things IgA can do.
Yeah.
And these are kind of one-offs, like in monocolonization settings and stuff like that. But it's interesting to think about, if an antibody promotes adherence of a bug, how does that feed forward loop ever end? Antibody is produced, bug adheres more, so there's more of it, more antigen, more antibody made. It's difficult to understand how those loops ever end. So it's really interesting for us to think about function of antibodies. And to do that, we need a system with more than one bug, a B cell we can track, and quite sophisticated sequencing approaches. And that's why it gets pretty complicated.
Absolutely. I think you're totally right that, the IgA they can be specific, but as a regulatory molecule, they're decorated with glycans, these non-canonical interactions. I think anti-microbe antibodies, not only in the gut, but we're thinking about them in the female reproductive tract. They're there. They're not just going to be there to clear things, but how do they harness things and keep niches
and how do
in
we
check?
study this outside of the environment where they become glycosylated and modified?
Yeah, right.
So I'm very excited about our future episode on things that Steph and Carla collaborate on.
Yeah, absolutely.
Yeah.
During the pre-show, you mentioned all of the ways where some of what we know about germinal centers can vary based on location and based on context of antigen delivery and all of those things. And so all of this is absolutely fascinating. How much of it is specific to the gut?
Yeah.
And how much of it is specific to, say, microbiome versus some kind of pathogenic infection?
Yeah, great question. Great question with not a lot of ends that can be tied, So the gut, the cool thing about the gut, beyond it just having a microbiome that we can live with per se and food going in all the time that we can live with, is that it's tolerogenic, autoimmune, and inflammatory, all at the same location, but usually not at the same time, but sometimes at the same time. So we can start combining inputs and then saying, okay, so we know we have a germinal center population that is existing, we'll call it in homeostasis, that's bug. Now what if I add in a pathogen? So then I start creating an inflammatory insult, and I say, what happens to those pathogenic ones? Those homeostatic ones, are they still there? Are they being overtaken? Is it the same structure? And what do we know about the selection dynamics here versus here? Then I can ask, okay, what if I start making this environment auto-inflammatory? And I can do that in the gut quite easily in a number of ways. I can add in stimuli, I can use specific mouse models, and I say, what happens to those homeostatic structures? One, what happens to the microbiome? And then what happens to how well we deal with other infections when we start tipping balances one way or the other? So it's quite useful to have a sort of baseline understanding of these sort of different scenarios. I haven't even touched on tumor because things get quite complicated there, but that's obviously a hyper-inflammatory environment, or a tolerant, or a exhausted, whatever you want to call it. But we wonder if cells in the gut being stimulated all the time by these sort of generic microbial inputs either become hyper responsive or dampened in their responsiveness because they become so blind to these signals. So it's a really cool environment to start messing around with the signals that are there and seeing how the cells respond. So we're super interested from the 2 research experiences I've had so far in understanding how that B cell gets rewired. Because clearly something happens from being a naive B cell to a germinal center B cell. Your wiring is completely reset so that your cytoskeleton works differently. Your signaling molecules work differently. You interpret force differently. And how that is regulated in these different environments, completely unknown. How it is even regulated in these kind of weirder structures like the Peyer's patch, the lung, great example, the skin, we're not sure. So there's lots of structural differences in these unique organs, and we don't know what it means for much, really. The subepithelial dome of the Peyer's patch is a perfect example, where the antigen in there might not be complex with another cell. It might be free floating. People have shown there's live bacteria in the Peyer's patch. It's kind of a crazy thing to think about. So then maybe all the stuff I told you about free floating antigen is completely moot in the Payer's patch, and we need to start rethinking how cells get turned on to antigen. Germinal center B cells are usually blind to free floating antigen, probably to stop us from making lots of harmful antibodies. But in the gut, maybe that's not the case.
Right. Wow. Well, so what your lab is working on now. So you started your lab when? What year was it? Were you a 2020
2
year?
years ago now.
Okay.
2 years.
2 years. Okay. And so tell us a couple, one or two things that you're excited about. I mean, you shared a little bit, but anything else you'd like to share?
Yeah, we're super interested in understanding signaling downstream of immunoreceptors. So might not be so much of a surprise that my favorite immunoreceptor is the B cell receptor. We study how it moves, how it signals, how it evolves, but we've become interested in a couple of other receptors too. So you can imagine the toll-like receptor and the other pattern recognition receptors are pretty important in the gut, and how these receptors start to talk to one another and what that means for whether a B cell enters a germinal center or not is really where we're becoming interested. And combining these knockouts with confetti, we're seeing some amazing results in what this means for germinal center selection dynamics. We're also pretty interested in early life, pretty interested in understanding how the system gets set up. We have looked at it so far, quite far down the line. So we're excited to see how that turns out. Hopefully we can unravel some of the complexities going on there. But yeah, mostly we're working on imaging a bunch of different kinds of B cells from the gut under all different circumstances, moving through to autoimmune malignant infection settings, as well as looking at any other kind of unusual germinal center.
Wonderful. Well, kind of one of our wrap-up questions that we had is what advice would you give yourself as a graduate student or as a early career scientist? What are some pearls of wisdom that you can pass down?
I truly think just don't think too hard about it. Don't think too hard not about the science, think very hard about that, but don't think, that's not my advice, to not think about the science. My advice is the career path, don't think too hard about it. Give it a go. Apply to everything you want.
Right.
Don't think about whether you can do it or not. You probably can.
Yeah. Just do it.
That's very cool advice.
I love that.
Just do it. Just do it. Why wouldn't you be able to?
Love that.
Yeah. Thinking about it too hard gets scary. So it's best just to apply, try your best, and do it.
And
that's a good
if
advice
you plan
for
too
grants
much ahead, then
too.
how are you going to be able to take advantage of super cool new techniques that you don't even know about yet?
Yeah.
Right.
Exactly.
Yeah, exactly. Or if you start thinking about all the other people who might get a grant except for you, it becomes scary to write. And I think people can read fear.
Yep. For sure. Yeah. I like that advice for approaching grants, things to apply for. It's like, just do it. I mean, you want to write it well, right? You don't want to annoy the reviewer and not write a good grant, but don't be, I think my thing too is don't be too personally attached to it. It can be hard, of course, but because then if you don't get it, it's like, okay, you're moving on. You have the next thing.
But when it's like applying to faculty positions or PhD programs or postdoc openings, why would anyone else get it except you? I think this is the mindset to go, why is anyone else better qualified than you? They're not.
Right.
Why is anyone else more qualified to be there than you? They're not. So I think that's the mindset to go in with, that you deserve to be there as much as anyone else.
Right. Beautiful. I love it. Cindy and Brianne, do you have any other questions for our guest today?
I was going to ask you, what were the first people that you brought into your lab? So as you're setting up your lab, this is a common question that I get for early career faculty. They're like, who should I hire first? Should I take a graduate student? Should I try to get a postdoc? Should I take a technician? Do I work in the lab? Can you kind of just give a little bit of a quick minute or two about your approach to starting your lab and how you built that?
I will disclaimer it by saying my previous advice perhaps is not the best advice for starting the lab. Do think about that one, because I did not. I don't know if I'm the best person because I just did it how I figured it out as I went along. But I would say I've received really good advice from both Gabriel and Daniel, where they advised to pick a postdoc very carefully, because that person is probably going to generate the very important early data coming out of your lab. So I was fortunate to find a postdoc early on who has been completely instrumental at getting the lab up and running. And leverage your whole network. Ask everyone you know to recommend people, and be really realistic and truthful about what you can offer them. And yeah, a technician is essential, I think. A technician to get the practical parts of the lab moving, because you're not worrying about biological phenomena for maybe a year. You're asking why your cells aren't growing.
Yeah.
So it's like
For
you're
sure.
not getting into like the major problems you had as a postdoc for a long time. You're like, why is our IACUC approval not come through? So that's the kind of stuff that a tech, a really efficient, motivated tech is really important for. Students are great, but students need hands-on training and mentorship, and you cannot not do that, because you see the results of that reflected in their data, because they don't know how to troubleshoot in the way a postdoc does. So I took on a student when I knew I would have time to train them, or when the rest of the lab was developed enough that there would be a hierarchy where they had people to go to for help, because I don't feel like it's the right thing to do to put a student in the lab who doesn't have anyone to go to for help. So you have to either make yourself available or someone else available. So the long and short of that is try and get a postdoc if you can, but make sure it's a postdoc you can communicate well with. I think, that's really, really important. A postdoc who is not exactly like you. Someone who can, I'm like a bit highly strung, so I hired a relaxed postdoc who could be like, everything's going to be fine, don't worry. And we run a good system together where I'm like, come on, and they're like, it's okay. Yeah,
it's awesome.
You mentioned that when you were doing your PhD, you had a plan that you wanted to come do a postdoc in the US. And so I wondered if you could talk about kind of what kind of differences do you see in science in the UK versus in the US? What's the system look like, and what was it like to adapt?
So plan is generous, thank you. I think it was more like a pipe dream, but I think there's a reputation in the UK of the US being really cutthroat. So like, you're going to go to a lab where 4 people are put on the same project and it's going to be miserable, and that's just not reality. The reality of science in the US, especially, I was at Rockefeller, so at a place like Rockefeller, is that you're going to be under some pressure and everyone's going to be working hard, because it's a great institution where people are doing really cutting edge science. So you go there because you're hungry to achieve, not because, so people go there knowing they're going to be working hard because they want to. They're really hungry for more than they had from where they came from. So I would say the main differences with the UK and the US is that in Gabrielle's lab, we were really limited only by our imaginations. We were sitting and truly thinking, right, how would we be able to do this experiment? And we had the kind of resource and people around us and facilities to just try it, give it a go. Whereas in the UK, there's lots of regulatory approval, which can be limiting. And there's much less funding around, I would say, in general. I didn't experience it at the Crick because we were centrally funded in like an NIH model, but I know that other people become limited by that. And in the UK, you're time limited. So the PhD is 4 years maximum. And I don't think that's inherently a bad thing, because there's 2 sides of the coin here. Some people get stuck in never-ending PhDs, and there sometimes you're out without a paper, which is restrictive. So I truly think, like, after being in both systems, that 5 years is the sweet spot, or some flexibility is good, but 7 or 8 years for a PhD makes it really scary to leave.
Right. Wow, excellent.
And that science in the US is very accessible, and it's a great place to achieve if that's what you want. But you don't have to work every weekend. You don't have to do any of that. You just need to show up, think everything is really cool, and try your best.
I love that. I think that's a good ending for us
Yeah.
today.
Yeah.
I think things are very cool. Be curious and try your best. I love it.
Yeah.
Well, excellent. I will pause in case anybody has any lasting thoughts, and then I'll close us out for
Yeah.
today.
Oh, thank you so much for having me. I've had such a
Yes.
nice time.
We loved having you, and that is Immune episode 74. Questions and comments can go to immune@microbe.tv. You can consider supporting the science shows of Microbe TV at Microbe TV /contribute. Microbe TV is a 501c3. So Cindy Leifer is at Cornell University at Cindy Leifer. Thanks, Cindy.
Thank you. This was really great.
And Brianne Barker is at Drew University at BioProf Barker.
Thanks. I learned a lot.
Yeah, thanks, Brian. And Carla Nowosad is at New York University, and we hope to have you back on in the future. Thank you so much for taking time today.
My pleasure.
Wonderful.
Thank you for having me.
And if you're curious, the music on Immune is by Steve Neal. And thanks for listening to Immune, the podcast that is infectious. We'll be back next month.